So now it's 11 o'clock, and today's event is a focus on Elicera and Elicera's full year report. But due to the nature of the company, our CEO Jamal Mosleh will dive into the most significant events during 2021 and take us through the latest events after the report period. So first of all, I would like to welcome Jamal Mosleh from Elicera, and I also want to welcome the audience today. And as usual on HCA platform, if you want to post some questions during the presentation, please feel free to do that in the right bottom corner, and then I will post them forward to Jamal during the presentation. So with those words, I will hand over the word to Jamal. Welcome, Jamal.
Thank you, Klaus. Thank you. So, as Klaus said, we recently released our year-end report. So I wanted to take the chance to talk to you guys about what we did at Elicera during 2021. I won't be talking too much about the figures and numbers and so on. I don't think it's that relevant for a company such as Elicera, but I can conclude that we had cash equivalents of 52.4 million year-end, and it's sufficient to last us until mid 2023 at least, according to plan and budget. So I will talk a little bit more about this later on, but I will start with a short presentation on the company for those of you that might not have heard that much about Elicera before. So it's a cell and gene therapy company focusing on immuno-oncology. And what we do is that we develop enhanced CAR T cells and oncolytic viruses. So we have quite a broad clinical pipeline or pipeline of drug candidates, of which one is in clinical stage. We have 2 CAR T cells under development and 2 oncolytic viruses. And we gene modify our drug candidates so that they can elicit a parallel immune response and a parallel attack by the patient's own T cells, meaning in parallel to the attack by the CAR T cells and the oncolytic viruses. We have an experienced board and management, so we have experience from immuno-oncology, we have experience from cell therapy, and we also have experience from patient studies, both with CAR T cells and oncolytic viruses. It puts us in a good position to develop these types of products. This is our pipeline of different drug candidates. As you see, the most advanced is ELC-100 in neuroendocrine tumors, where we are currently conducting a dose escalation study. So we're halfway through that study. We are also developing ELC-201, which is a next generation oncolytic virus applicable for treatment against most tumors. And we have also genetically modified this to elicit a combination of 3 mode of actions in one product. And 201, 301, and 401, all of them are including the ITANK platform, which is a universal CAR T cell technology platform compatible with any CAR T cell therapy for a multi-targeted attack on solid tumors. So we're using this also for this oncolytic virus, but the patent and the main area of focus for us with the ITANK platform is the CAR T cells. In that area, the CAR T cell area, we are developing a drug candidate which we call 301 in blood cancer. Initially, we will treat patients with B cell malignancies. Our ambition is to start our first clinical study this year. And then we're also developing 401, which is in a range of solid tumors, not all of them, but a range of them. And initially, we will treat patients with brain tumors. Our business model is based on out-licensing both our ITANK platform, but also our different drug candidates. And the strategy for out-licensing and finding partners for our drug candidates is to do that after we have completed at least clinical phase 1/2 studies. The reason for this is that we believe that we can create most value and minimize risk in this way before finding a partner. The ITANK platform, however, being that it's fully developed, we are looking for partners for this platform already today. And as I said before, it's a technology that we think is applicable universally across any CAR T cell. So it can solve two of the major problems that basically any CAR T-cell developer is facing in the treatment of solid tumors. And that's why we think that this platform is very relevant for any developer of these types of products. We are developing both niched and more broad products in both categories, both CAR T-cells and oncolytic viruses. So ELC301 and ELC100 are more restricted to certain indications, while 401 and 201, 401 being CAR T-cell and 201 being oncolytic virus, are applicable against a range of different tumors. So there's a broad potential to find different licensors for these drug candidates that we are developing. This is a picture of our 3-year clinical development plan. You see that we already have an ongoing clinical study, and we expect to start another one at the end of 2022 in B-cell malignancies, our first CAR T-cell study. And then later on next year, we expect to have actually 4 clinical studies ongoing in parallel that will be completed in different stages. And the cash that we have today, as I said in the beginning of this presentation, is sufficient to give us a runway at least until mid-2023. So to take you through the significant events that we had during last year, I'm proud to say that we were able to reach all of the goals, the major goals that we had set for the company. We listed Elicera on Nasdaq First North Growth Market on June 11th. And in connection to that, we also completed a oversubscribed new share issue that generated proceeds of 55.1 million after costs. So we're in a good position now to continue developing our drug candidates according to plan. We also submitted a patent application to protect ELC201, which is a next generation oncolytic virus that I just described to you. We received a ATMP classification from the European Authority, EMA, for the oncolytic virus that is in clinical studies today in neuroendocrine tumors. And this is important since it makes the pathway more clear towards market, the regulatory pathway for this specific product. We also, basically all of us, more or less in the management and in the board of director, also actually increased our shareholdings after completion of the new share issue. We purchased additional shares. We added Gunilla Enblad, which is a key opinion leader in the field of CAR T cells and B-cell lymphoma, as scientific advisor to the company. She's also the one that will be principal investigator for the upcoming 301 study that is to be initiated this year. Importantly, we also presented ITANK proof of concept data at 2 conferences, the ESGCT conference and also the PEGS European Summit conference. So this data, basically to summarize it, we were able to show that we compared the efficacy of CAR T cells that have been enforced with the ITANK platform, and we compared it to conventional CAR T cells that did not include this platform. And we could show that we can increase the efficacy against cancer with the ITANK platform, regardless of the choice of CAR molecule, regardless of choice of tumor indication, and regardless of choice of animal model that was used. And this clearly indicates then that this platform is universally compatible with any CAR T cell under development. Finally, last year, we also entered 2 agreements with 2 contract manufacturing organizations, BioNTech and Baylor College of Medicine, to produce virus vectors for ELC401, the CAR T cell, and also ELC201, the oncolytic virus. So that was important activities. And we also, after 2021, meaning in January, so this year, we've had 2 significant news. One is the acquisition of a patent from Immunicum to strengthen our IP protection for ELC100. This patent is approved in the US. And this patent was reacquired from Immunicum, so it was actually sold to them in 2014. And in that agreement, we had mutual royalties and milestone payments for both parties. that are now terminated. So we believe that the strengthening of our IP position for ELC100 and the fact that we don't have any mutual royalties anymore with regards to the old agreement puts us in a better position to negotiate with a potential partner for ELC100. And then finally, we've also reported that we were approved a grant of 5 million Swedish kronor from Vinnova to develop a automated CAR T-cell manufacturing process. So CAR T-cells are very complex and time-consuming to produce, and this project to create an automated CAR T-cell manufacturing process is, of course, to make that process more robust, more efficient, and to be able to not take as much time. So we've had a very successful and exciting 2021, and we have perhaps an even more exciting couple of years ahead of us. So during 2022, we are expecting several important news revolving the ITANK platform, starting with a publication in a scientific journal of our preclinical proof of concept data. This is important since we believe that it sends an important message both to academic and to potential commercial partners around this technology. We also expect to have an increased pace in recruitment of patients for the ongoing ELC100 study, with the ambition to have completed the dose escalation phase during this year. We also expect to have completed successfully GMP manufacturing for both 401 and 201 in order to pave the way for a quick initiation of future clinical studies. And of course, to initiate our first CAR T-cell study in ELC 301 in patients with B-cell malignancies. And then finally, we already have ongoing partnering efforts, and we expect to continue these partnering efforts with the ambition to secure our first commercial partner for the ITANK platform during this year. So thank you so much, and I'm opening up for questions.
Yeah, thanks a lot. Very good presentation, Jamal. There's a couple of questions popping in here, but you released very good proof of concept data on the ITANK platform, and after you released them, even though you will release them in scientific journal as well, do you already now see an increased interest for the platform, Jamal? And have you so far signed any agreements?
Yeah, so there's no agreement signed yet, but yes, there has been an increased interest in Elicera and the ITANK platform, and we expect that to increase even more, as I said, once we have the publication also out there. So I won't speak more in detail about this, but rather inform the market, of course, when there's more to inform, if there's any news and developments in that area. Of course, I understand, but it was quite versatile, the possibilities with ITANK, as you also mentioned in a couple of slides ago. And then you also broaden your spectrum within the value chain for a biotech company by building your own GMP production. Will you offer this production capacity for other players in the field and look into partnerships? Because I know that's a major problem for many producers to find the right place to produce cells for immunotherapy. Yeah, so it's a good question and important for me to also clarify then. So the project that we have received grants from Vinnova here of 5 million Swedish kronor is not to actually establish our own GMP facility. That costs a lot more, and it's a much bigger process. So this is in order to automate the process of creating the actual CAR T cell. Okay. So when the CAR T cell therapy is actually, you can say, produced in two separate steps. Actually, more steps, but in two major areas. For one, you need to create the vectors, and then you need to use the vectors to actually construct the CAR T cell. So the automation is with regards to the second step here, and it's a smaller but nevertheless an important step to do, and it's something that we hopefully will be able to also generate some IP protection around. So if this project is successful, and it's possible, we will submit a patent application to protect that process. And in that case, yeah, it will become an asset of Elicera that could potentially also generate commercial interest, as you say. But we wouldn't be completely unique in this. So different companies and organizations have their own, not always, but sometimes they have their own solutions on this automation process. Otherwise, they need to acquire it or license it from somebody else. In this way, we would be able to do it on our own. We would own it, and it would be tailored for our specific needs. So that would be the good thing here.
Yeah. Is it more complex to do step 2 than step 1, because you entered a cooperation with BioNTech in respect of the vectors, if I remember right?
Yeah, I mean, you can say it like you can see it in the sense that creating or producing the vectors is like producing the raw material for the CAR T construct later on. But the CAR T cell is, of course, based on the patient's own T cells. So once you have the virus vectors, then you need to extract the patient's own T cells and tailor make the specific CAR T cell therapy for each specific patient using the virus vectors. So in that sense, creating the CAR T cell is a little bit more complex, I would say.
Okay, and in that sense, it makes good sense that you have that in-house then.
Yes.
For sure. Well, then there's a question here about the current situation with the pandemic. If you experience any disturbance in relation to recruiting patients for your most progressive study?
Yeah, so what we're trying to do now is to speed up recruitment both in Sweden but also abroad. So we're looking at the possibilities of opening up new sites in Germany. And yeah, for sure. I mean, the pandemic has made it all of these processes with interaction with different regulatory authorities. It takes a little bit more time, for sure. But we're hoping to be able to initiate the activities that we're planning in order to speed up recruitment. And of course, as soon as we have some news on that, we will report it.
Great. Thanks a lot. Thanks a lot. Well, let me see if there's any other questions right now. There's no questions. And you briefly touched upon the finance situation as well, and there's not much to say. You have funds sufficient, you mentioned, until the mid-2023, and that includes the 2 most progressive projects you have, I could imagine, Jamal. So ELC 100 and the 201.
Yeah, so we have cash actually that lasts a little bit into also second half of 2023, and this is based on the budget that we previously set and that we're following quite well. Now we've received the 5 million Swedish kronor from Vinnova, that grant, and that would also actually make it possible for us to stretch the runway somewhat, not very significantly, but it puts us in a little bit better position, of course, because this project would have been concluded anyway. So it saves us some funding here. Yeah. Regarding our burn rate, I would say that we actually have a little bit higher burn rate during 2022 than 2023. And this is mainly due to the production activities that we have. So what we did late last year is that as I said here, we initiated the production of both 401 and 201. So 100 is already in clinical studies, 301 is next to enter patient studies, but 401 and 201, we don't want to lose time, and we want, of course, to be able to enter patient studies as soon as possible. So for that reason, we initiated GMP production of those drug candidates. The process was initiated last year, and we did that in order to reach patients as soon as possible, and that, of course, takes some cost during this year. Yeah. So the ambition is to have concluded that during 2022. That's why the run rate is a little bit lower during 2023. Yeah,
of course. And if you then receive if you sign up for partnerships on your ITech platform, I could imagine that is on top and then stretch. Well, then you can wait with raising money maybe to later 2023.
If it generates an upfront payments, yeah, for sure.
Yeah, good. Well, I think so far Yeah, there's a question about how much you will raise, but that's difficult to say right now, I could imagine. But
yeah, so the potential next raise is so far ahead in the future. It's not something I want or can't comment on at the moment.
Of course, Jamal, I understand. But with that said, and a really nice presentation, I don't think there's further questions to start with here, Jamal. We're really glad to host you today, and really glad for the presentation. You run through the most important things. And for the audience, if you have any questions whatsoever, you find Jamal's email here, and you're also allowed to post the questions to us in HC Andersen, and we will contact Jamal. So with that said, I would like to thank all the audience for attending today, and I will also thanks Jamal for a very good presentation. Thanks a lot.
Thank you so much, Claus.
Yeah,
thank you.
Enjoy your day. Thanks a lot. Bye
Thank
bye.
You. You too. Bye bye.